1 million. It's a staggering number, right? Well, right now, that number represents the massive energy efficiency gap we're hitting in modern computing. As we push artificial intelligence further and further, we're literally hitting a wall of heat. Modern AI data centers are devouring gigawatts of power, needing actual rivers of water just to keep everything cool. But then, think about the human brain. It handles infinitely more complex tasks, parsing context, feeling emotion, using logic, and it does the whole shebang on roughly 20 watts of power. That means our current AI setup consumes about 1 million times more power than that incredibly efficient, squishy organ inside your head. And honestly, this energy gap, it's the fundamental bottleneck of our entire era. To tackle this massive energy crisis, a team of researchers from Princeton University, led by Tianming Fu, James Sturm, and Kumar Matunjay, decided to completely flip the script. Instead of trying to force rigid silicon to act more like biology, they figured, why not just use actual biology? So they've physically merged living brain cells with advanced flexible electronics to create a programmable living computer. No, this isn't some sci-fi movie plot. It's a monumental breakthrough recently published in Nature Electronics, and it's totally reimagining what computer hardware can actually be. So for today's explainer, we're going to break this all down. We'll start with the AI energy crisis, then introduce you to the living computer, take a look inside its 3D scaffold, see how on Earth you train a biological network, and finally, dive into the mind-blowing medical applications and the ethical risks. Starting right off with the AI energy crisis, let's talk about the power bottleneck. To really grasp this bottleneck, we have to look at how traditional computing works. It relies on something called the von Neumann architecture. Basically, it's constantly shuttling data back and forth between memory and processing units, which just bleeds energy at every single step. And that's exactly why we're seeing this exponential explosion in the energy needed to run modern silicon-based AI. The biological brain, on the other hand, is a masterclass in efficiency. It processes and stores memory in the exact same spot, the synapse, and it only fires when it absolutely needs to. This natural sparseness is the ultimate blueprint for saving energy, which is exactly why scientists are pivoting away from rigid, overheating silicon GPUs and diving headfirst into the soft, wet, incredibly efficient realm of biology. Which brings us to part 2. Enter the living computer and the building of 3D biological networks. Now, going back to that groundbreaking Princeton publication where they unveiled this microscopic biological processor, you're probably asking yourself, can we actually call a literal jar of brain cells mixed with wires a computer? I know, it gives off major Matrix vibes, but incredibly, the answer is yes. It works through a concept called reservoir computing. Essentially, the highly complex biological tissue acts as a reservoir that takes input signals and transforms them into recognizable output patterns. The absolute key here is that this 3D biological neural network, or 3D BNN, doesn't just sit there passively monitoring cells from the outside, it actually integrates with them. By taking tens of thousands of living neurons to act as the active processors and using a microscopic metal mesh as the scaffolding and wiring, these researchers have built a hybrid computational system that can actually perform directed, programmable tasks. And what's a real game changer is how different this is from older methods. For decades, scientists relied on flat 2D microelectrode arrays, basically just smearing cells onto a rigid petri dish. But here's the thing, the brain isn't flat. Those older 2D cultures had terrible spatial resolution, very limited connections, and honestly, they just die off in a matter of weeks. But the Princeton team's new 3D micro-instrumented network uses a flexible volume approach. It allows for full spatial integration, and amazingly, it stays functional and stable for over 6 whole months. So how did they manage that? Let's move to part 3 and peek inside the 3D scaffold to understand this inside-out approach. To get that incredible stability, you can think of the structure kind of like a garden trellis. Instead of forcing these neurons to sit awkwardly on top of a rigid board, the researchers built a flexible microscopic metal mesh and coated it in an ultra-thin epoxy polymer. The genius of this is that the epoxy's elasticity is a perfect match for the softness of actual brain tissue. Because there's no harsh mechanical mismatch, the neurons don't freak out, they don't trigger a damaging immune response or form scar tissue. They just naturally grow around and straight through the mesh, treating the electrodes like they're just a normal part of their own environment. And because that mesh is so incredibly welcoming, researchers were able to track 4 distinct phases of growth over half a year. First, you have adhesion, where the cells quickly anchor themselves to the epoxy. Next up is expansion, usually between weeks 2 and 4, where you get this massive explosion of synapses forming. By month 1, things settle into maturation.
and the network actually starts showing synchronized, spontaneous activity, exactly like a real brain. Finally, we hit long-term plasticity. For 6 whole months, the network continuously reshapes itself, giving scientists a front-row seat to the ongoing evolution of a living, functional neural web.
Okay, moving on to part 4, training biological networks, or how we actually program these cells. So you've got this stable mesh set up. The next obvious and honestly fascinating question is, how do you actually train a jar of brain cells? Obviously, we can't just plug in a USB cable and upload some Python code. We have to speak the literal language of biology to get these organic cells to recognize patterns and do our computational heavy lifting. The secret here is leveraging something called synaptic plasticity. By feeding really specific, carefully timed electrical pulses through the metal mesh and directly into the tissue, the scientists were able to artificially trigger long-term potentiation and depression. In plain English, they intentionally strengthened certain connections between specific neurons and purposely weakened others. They effectively programmed the biological network to reliably recognize distinct electrical pulse patterns and then used a machine learning algorithm to read the signals coming out of the mesh. Boom! They successfully trained a living network to compute. And when you stock this up against other cutting-edge hardware, the difference is night and day. Sure, neuromorphic silicon chips are a big step up from your standard GPUs, but they still fall short. We've also seen some amazing stuff from places like Northwestern University, creating artificial neurons out of organic semiconductors that run really efficiently on ion dynamics. But this 3D BNN approach, it hits the absolute maximum level of biological energy efficiency. Why? Because the power needed to maintain all those connections is provided by the cellular metabolism of the living tissue itself. It is, quite literally, the ultimate biomimetic AI. Which brings us to our final section, healing brains and ethical risks, exploring both the medical and moral horizons. Now, beyond just saving a ton of electricity for future AI, this technology unlocks some truly staggering medical possibilities. Because this flexible mesh totally avoids triggering that awful scar tissue, researchers can actually grow these 3D networks using patient-derived stem cells. That means creating real-time functioning models of diseases like Alzheimer's or Parkinson's and testing drug efficacy directly on the network's actual synapses. Even wilder, this paves the way for next-generation neuroprosthetics, literal implants that can sit seamlessly inside the human brain, speaking its native electrical language to bypass damaged tissue and restore lost functions without the body rejecting them. But we do have to look at what's coming on the horizon. Right now, we're dealing with tens of thousands of cells in a tiny distributed network. But as we inevitably scale this up, from thousands to millions and eventually billions of integrated neurons, we really have to pause and think about the status of the living substrate we're creating here. This whole newly emerging field is known as organoid intelligence, and it physically blurs the line between organism and machine, forcing us to confront some pretty uncomfortable questions about awareness. So, I want to leave you with this provocative thought. As we eagerly harness this rudimentary sentience and biological efficiency to power the future of our AI systems, what kind of moral frameworks are we going to need? If a biological computer begins to exhibit massive-scale self-organization, how do we make absolutely sure our advanced living computers aren't actually experiencing distress? It's a fascinating and, yes, slightly terrifying frontier, and it is unfolding right before our eyes. Thanks so much for joining me on this explainer, and remember to keep questioning what's possible.